I should start fast. So welcome back. This is the first thesis defense I've been doing in a really long time. I'm super excited that we're in a phase where we can do this in person in this moment. So I'm going to say a couple of brief words on the Nancy's, and then we're going to let Heather do the actual event of the day. I've been trying to tell Heather that this moment is just a ritual, right? That her growing into being the independent scientist that she is a story that started a long time ago, like a decade ago, and is not stopping today or really even changing very much today. Tomorrow will be a lot like yesterday. And there's just no question that Heather has already earned a PhD. This is not a test. It's a celebration. So I'm really glad you guys are here to mark with me this moment of epochal transition as we recognize everything Heather's accomplished in the last 8 years at MIT. And you won't get to see it, but I just want to let you know that her thesis document, the written thesis, which is going to be submitted to MIT, is incredible. It's the kind of thing you imagine that a PhD would be, right? It's a huge body of work that starts with fundamental open questions, which we genuinely don't know the answer. It then goes through a long process of technical innovation, incredible technical innovation, in how the signals will be measured and how they will be analyzed. Then a phase of ridiculous green thumb data collection, the likes of which one can only imagine, and then deep and clear and thoughtful reflection on what this means about the nature of the mind. And we grant PhDs for all different collections of work, but it's what you aspire to, right? To see a piece of work that big come to its conclusion the way that it does. So I recommend that you look at it. I always thought no one ever read PhD theses, but then Heather read mine. So please feel free to get my revenge by reading hers. It's totally worth it. And also I can see that when she reads other people's theses, they serve as examples to her of what is possible. And I really believe that her thesis will serve as that kind of example of what is possible to many generations to come. So it serves you right that that's a public document. And I'll just get this up to Nancy.
All right, so welcome everyone. As Rebecca has already made clear, Heather is a marvel. She's an inspiration. She is a force of nature. It has been my incredible privilege to get to work with Heather and with Rebecca, who found her, identified her talent, and trained her to launch the amazing research program you'll hear about today. So Heather's work addresses some of the most fundamental questions that humans have ever asked themselves concerning the origins of the human mind. And the particular version of this question that Heather's work addresses is the developmental origins of the functional organization of the cortex, a topic very dear to my heart. The standard work on this field, it's been going on for a long time, has been to scan kids age 4 and up. And that's plenty hard, and they've made some progress, but I think that approach doesn't really get at the core issues. 4 years old is just too old. Too much has happened by then to really find out about the core issues of the origins of the mind. So then a bunch of years ago, Rebecca and Ben Dean did some groundbreaking work, scanning awake infants for the first time while they looked at complex real-world stimuli. That was exciting and heroic and promising, but it left me wanting more. They found these activations, but it wasn't totally clear what they were or how they related to the adult versions of these things. And yet at the same time, I doubted whether anybody would ever be able to get beyond that because of the huge challenge of getting high quality data from infants. So that was when Heather came along, fearless and determined to push this nearly impossible research program farther. So for years, Heather worked with Rebecca to improve the methods for scanning infants. She designed her own new infant coil. She designed and built in collaboration with the headphone company Sensometrics, her own infant headphones. She spent a lot of time measuring the degree to which different kinds of hearing protection would attenuate the sound to make sure that the scanning was safe for infant ears. And that was really important because it enabled her to use adult pulse sequences in her functional MRI scanning. And that meant that instead of, well, that the brains actually looked like brains instead of mashed potatoes. And then after all that, Heather scanned an absolutely insane number of infants, as you'll hear. So somewhere in there, Rebecca and Heather very generously invited me to kibitz with them on this exciting work that they really launched. And I was intrigued, but still skeptical. And so to get a sense of what was entailed, I decided to watch Heather scan an infant. So I went downstairs and a train wreck was unfolding. The kid was screaming, the parents were nervous. It was awful. For 20 minutes, I watched this happen. And the whole time I'm thinking, when is she going to call this off? This is just intolerable. I mean, I just thought that it was ludicrous. But I sat there cowering in the corner. And after about 20 minutes, suddenly, the whole time, Heather's calmly bouncing the infant and reassuring the parents as if everything's fine when it was clearly not. And then suddenly the infant shuts up, the parents smile, the infant goes into the scanner, and Heather gets the data. So the data that Heather will show you today are about the hardest won data I've ever had any role in watching. A final note to be mentioned is that Heather did all of this while single parenting her remarkable daughter, Hannah, something very few graduate students attempt. And so kind of like Ginger Rogers, who danced every step that Fred Astaire did, but backwards and in high heels. Without further ado, Heather.
Hello, everybody. I know most of you, so it's okay for me to say that I knew I was going to be very embarrassed by the things that Rebecca and Nancy would say. I'm a little bit more embarrassed than I anticipated. So I'm going to do a little bit of payback and say some incredibly flowery things about my mentors. So for those of you that don't know, Rebecca and Nancy are extremely decorated neuroscientists that virtually created entirely new fields to better understand the mind and brain of humans. And they're a huge inspiration, not only to me, but to many people across the world, especially women that want to be scientists. But grad school has been really hard for me, not just professionally and intellectually, but personally. I've had a lot of struggles that I've had to overcome. So in addition to teaching me about science, Rebecca and Nancy have also given me many life lessons. And I'm just going to give you a small example of those right now. From Rebecca, I learned the importance of generosity. With my time and resources, and the way I interpret other people's intentions and behavior. That's construction, in case you're wondering. But also generosity to myself. From Nancy, I learned balance. She taught me that the bad stuff is going to keep on happening, but the key is to leave myself enough room so that when it does, I don't have to give up the things I love, like thinking about babies, brains, and minds. And both of them have provided me with incredible resources and opportunities to spend the last several years thinking about infants' minds and brains. And so I just want to say thank you for whatever things that I've said that you said that I did. I couldn't have done any of it without your support. All right, so let's go on to the talk now. As soon as I stop shaking. All right, so for those of you that don't know, this is a cartoon image of a brain. On the left is the lateral surface of the brain. On the right is the ventral surface. And we have parts of our brains that are highly specialized for different functions. There's a part of our brain that responds more to faces than any other visual category. and a different part of our brain that responds more to bodies, a different part of our brain that responds more to scenes. And if this is starting to sound like a laundry list of selective areas, it is a little bit, but it'll make sense in a little while why I'm telling you all of these things. There's also a part of our brain that responds more to speech than any other sound, even if you don't know the language. A part of our brain that selectively responds to music, and parts of our brain that are specialized for processing language, but only languages that you know. And perhaps most surprisingly, there's a part of our brain that's specialized for when you think about the thoughts of others. These discoveries are a few of the discoveries that my advisors made during their prolific careers, and which inspired me to start wondering, well, what is the starting state of the human mind? To answer this question, in my research, I used fMRI to study infants' brains while they listened to sounds like music and speech and watched colorful real-world videos. Today, during my talk, we'll listen to some construction noises. And I'll first ask, do infants' brains have music and speech selective responses in non-primary auditory cortex? Then I'll move on to vision and ask if infants have face, scene, and body selective responses in the same parts of the brain that adults do. The third question I'll address is whether cortical face responses emerge in series or parallel. And then I'll leave you with a few final thoughts about emerging minds. All right, so first, do infants' brains have music and speech selective responses? So let's talk about the adult brain for a second. One of the fundamental organizing principles of adult primary auditory cortex is tonotopy. There are regions outlined here in black that respond more to low frequency, sounds that have low frequency information, and regions here outlined in white that respond more to sounds that have high frequency information. This is called tonotopic organization. And if you play humans a bunch of sounds organized by category and then measure the response to these categories, you won't see any structure that resembles the category structure. Rather, it resembles the amount of low frequency or high frequency content in each of the sounds. Outside of primary auditory cortex, there are category selective responses. There's a region that responds selectively to speech, which means that the category speech explains the response of this region more than any of the spectral-temporal statistics of the acoustic sounds. And another region that responds selectively to music. Again, the category music explains the response in this region better than any of the statistics of the sounds. The question I want to ask is, do infants' brains respond to music and speech in the same way? So to do this, I created infant-friendly stimuli, specifically recording mothers while they speak to pictures of their children in a recording booth. Recordings were 18 seconds in duration with a clear onset and a clear end. And the mothers were looking at pictures of their infants while these recordings were made. I also selected a handful of lullabies that would be unfamiliar to infants in a New England context. I hired multiple musicians to play these lullabies, and I instructed the musicians to play them specifically in the frequency range of IDS by giving them the appropriate music. And importantly, the instruments we selected emitted noise from a single source so that it would be similar to singing. And again, the recordings were 18 seconds in duration with a clear beginning and a clear end. All right, so the first stage of sound processing in auditory cortex is that, is that the sounds hit your ear, right? So when a sound comes in, it hits your ear and it activates your cochlea. The cochlea then decomposes that sound into a waveform, the sound into frequency and power. So I'm just going to plot the frequency and power of each of my stimuli on this graph. The frequency is going to be on the x-axis and power is going to be on the y-axis. And music will be plotted in blue and speech sounds will be plotted in red. All right, as you can see here, there's quite a bit of difference between music and speech sounds, indicating that natural speech and natural music sounds have low-level acoustic confounds. So if the brain differentially responds to these two categories of stimuli, it might just be because of these low-level confounds, not because the brain is preferentially responding to either music or speech. So I'm replotting here just the mean of the two categories. And what we're going to do is we're going to iteratively change the amount of power in each stimulus at each frequency so that across the categories, they're matched. In other words, we're going to match the amount of excitation of the cochlea between the two groups of stimuli. And so that's what we did here. You can see it was fairly successful. We had pretty well matched stimuli based on the way they activate the cochlea. But if you're like me, you might be like, well, won't that make the speech and music unintelligible? So I'm going to play some sounds for you now. Hopefully there won't be any construction so you can actually hear them because they'll play quietly. This is an example of an excitation matched speech sound.
Hey, silly noodle, who you giving a hug to? Are you giving a hug to that monkey? You giving a hug to your friend the monkey? Oh, now you're giving him a big kiss. Are you chewing on that monkey's face? That's so silly. That's so silly. You like that monkey a lot, huh?
Okay, so as you can see, it has a lot of speech structure. You can clearly tell it's infant-directed speech. And now I'll just play for you an example of a music stimulus. Again, you can see that the music stimulus is still fully intact. It's recognizable as a lullaby, maybe to some of you. And for those of you that are even more proficient, you might have recognized it as a flute. For those of you that know Dana Bobinger, you might know that she was the one that played it. But in addition to frequency and power, sounds are processed according to time, so temporally. So next I'm going to plot a cochleogram for you of each of these sounds that I just played for you. On the x-axis now is time, on the y-axis is frequency, and the color indicates the amount of power at each frequency at each time point. So as you can see, these two sounds are not matched when you talk about temporal modulation. Temporal modulation is processed by primary auditory cortex. And I can't match these two sounds for temporal modulation because if I did, I'll lose the structure. It will no longer sound like speech or music. So instead, what we're going to do is create model matched versions that are matched on the spectral-temporal statistics of the stimuli. So I'm going to show you a model matched version of the speech sound. This is the cochleogram. As you can see, it looks pretty similar. So it seems like it will activate primary auditory cortex. This is it. As you can see, it no longer has any of that acoustic structure that you would have recognized in the speech sounds. But again, it has the same, it's really well matched for the spectral temporal statistics. And we did the same thing for the music stimuli, which I'll again play for you. Okay, so I hope you heard that. I'm sorry, it's really quiet. I can't turn it up any louder. But the main point is that the model matched versions, although they have the same spectral temporal statistics, they don't sound like typically occurring speech and music sounds. All right, so what would our hypothesis be for how these sounds would elicit activation in infants? Well, one possibility is that infants' cortex won't yet be tuned to anything beyond frequency and power. This would predict a cochlea-like response in primary auditory cortex, such that you would see that a similar response to music, plotted here in purple, model match music plotted in teal, speech plotted in pink, and model match speech plotted in yellow. Alternatively, it's possible that infant primary auditory cortex has spectral-temporal tuning. This would predict that voxels that respond highly to music will also have a high response to model matched music, and voxels that respond highly to speech will also have a high response to model matched speech. The third possibility is that infants, like adults, have category selective responses. That is, they have responses to music that can't be explained by the spectral-temporal statistics of the stimuli, and they have responses to speech that also can't be explained by the spectral-temporal statistics of the stimuli. All right, so let's actually test this. All right, so I combined data sets across 2 different coils, an adult 32-channel coil and our custom infant head coil, which I'll show you a picture of in a minute. And we swaddled infants and applied hearing protection, which Nancy told you all about already, that both played sounds to infants and attenuated the scanner noise. And then we put them in a custom head coil that had a size adaptive function so that it could accommodate different head sizes as well as the headphones. For this study, we recruited 50 infants. We were actually able to get data from 36 of those infants, and the infants were between 2 and 12 weeks old. Also, in case I forgot to mention, these infants were sleeping. To be included in the analysis, the volume to volume motion had to be less than 2 millimeters or degrees of motion. There had to be at least 48 consecutive low motion volumes, and at least 2 subruns for the ROI analysis. So our primary analysis of interest that we're going to talk about today is a functional region of interest analysis. So here I've plotted a typical motion run. On the x-axis is the number of volumes, and on the y-axis is the frame-to-frame displacement between volumes in terms of translation. As you can see, there are some areas where there's not much motion, but there's some areas where there's quite a bit of motion. And so we want to get rid of all of the bad data that's been corrupted by motion by selecting out the low motion portions and creating what I've termed subruns. As you might imagine, different infants are going to move different amounts at different times. So we're going to have a variable number and duration of subruns from each subject. But I'm going to run through one subject for you and show you how we do an ROI analysis. So for example, this subject has 3 subruns. So we'll take one partition of data and leave one subrun out and do a voxelized general linear model to compute the response to music that's greater than the response to speech, and do the same thing separately in the left out subrun. And then we're going to find an area of interest. In this case, I've highlighted the auditory cortex, and find this top 5% of voxels within that parcel that respond more to music than speech. Then in independent left out data, we'll extract the response to those voxels from music, model matched music, speech, and model matched speech. For example, this might be what one infant's response looks like. The first question we're going to ask with this kind of an analysis is, can we activate primary auditory cortex in sleeping infants with our stimuli? First, I'm going to show you a group random effects analysis, which just averages the voxel response across all subjects. And you can see here, there's an activation in primary auditory cortex that's quite small, appears to be left lateralized, but it's significant. So we might want to know though, do all of these stimuli activate primary auditory cortex? And so we'll do our ROI analysis with a contrast of all stimuli sounds over the scanner noise alone in all of our infants. So all 36 of the infants that contributed data. And on this plot, I'll plot the response magnitude on the y-axis. And you can see here, we do indeed significantly activate primary auditory cortex with all of these sounds? So yes, we're able to activate primary auditory cortex in sleeping infants. But what's hidden in these data a little bit is some infants have really nice activation, primary auditory cortex activation. As you can see, this infant is 3 weeks old and has really lovely bilateral activation along Heschel's gyrus. But this infant is about 6 weeks old and seems to have no activation whatsoever. And so about 19 of our, well, not about, exactly 19 of our subjects had significant activations in primary auditory cortex, while 17 did not. And so we want to know, is there any obvious reason for this difference? And we didn't find any differences between these two groups based on motion or based on their age. But we did find a slight trend that the infants without significant activations had a little bit more data than the infants with significant activations, which is surprising. So next we're going to look at just the infants that had significant activations and do an overlap map to see what parts of the brain are activated by these sounds. And when you do that, you can see nice bilateral Heschel gyrus activations, which confirms that we are indeed, at least in a subset of infants, getting really nice auditory activations. But I told you at the beginning that we're interested in the response to speech and music. So let's look at that map. So first we did the group random effects analysis. And this time we're only going to look in the infants that had significant auditory activations. Again, these infants range in age from 2 to 12 weeks. I'm going to show you the left side of the brain on the left and the right side of the brain on the right. And here you can see there's a significant activation to music, but it's ventral to where we would have expected in the auditory cortex. And if you look at individual infants, we do get responses to music in both the left and right hemisphere, but we also get responses to speech in both left and right hemispheres. So this is promising because it's showing us that we are getting our music and speech responses, but it doesn't tell us whether or not these responses are selective to music and speech, or if they're just responding to the spectral-temporal statistics in these stimuli. So to answer that question, we're going to do our ROI analysis again, but this time looking at music over speech and speech over music. And just to remind you of the hypotheses, one possibility is that infants' brains will respond to the spectral-temporal statistics of the stimuli, but not prioritize music or speech per se. And alternatively, it's possible that there'll be a music selective response or a speech selective response. So we're going to do our ROI analysis in primary auditory cortex of music over speech and ask if infants' brains respond selectively to music. Again, we're only going to look in the infants that we had significant auditory activations for. Here's the response to music. It's significantly greater than the response to model matched music, suggesting that the spectral-temporal statistics of these stimuli are not sufficient to explain music responses in infants. It's also significantly greater than the response to the other categories. For speech, so yes, music responses are not explained by the spectral-temporal statistics of the stimuli. Now we'll ask the same question, but looking at speech over music. Here's the response to speech. It's not significantly different than the response to any other category, which we found surprising given we found speech over music responses in some individual infants. So next we did an exploratory analysis collapsing across all of the infants. And here's the speech response. As you can see, in this analysis, the speech response is significantly greater than the response to all other categories. So do infants' brains have speech selective responses? I would say it's unclear. All right, so do infants' brains have music and speech selective responses in non-primary auditory cortex? Maybe. All right, let's move on to do infants have face, scene, and body selective responses in FFA, PPA, and EBA? So adults have category selective responses in ventral temporal cortex. There's a region called the fusiform face area, or FFA, that's highlighted here in purple. It responds more to faces than any other visual stimulus. An area called the parahippocampal place area, highlighted here in green, that responds more to scenes than any other visual stimulus. And an area called the extrastriate body area, or EBA, highlighted here in pink, which I'm sure you've guessed by now, responds selectively to bodies. All right, so ventral temporal cortex, I'm going to show you a little cartoon of an infant brain. The FFA usually lands right about here in the posterior kind of lateral part of the ventral temporal cortex, where as the PPA is more anterior and medial than the FFA. So this has led to a hypothesis that category selective responses develop as a result of overlaying low level visual feature maps, like retinotopy that goes from foveal to peripheral, spatial frequency from low to high, and curvilinear to rectilinear properties. These feature maps are overlaid on one another in VTC, and with lots of visual experience, with something like faces, people develop a face-selective response in the area that responds maximally to images with low spatial frequency content that are foveated and have lots of curvy features. And then with lots of visual experience with scenes, people develop scene-selective responses in area of cortex that respond maximally to rectilinear items that have lots of high spatial frequency content and often fall in the periphery. To test this hypothesis, I used fMRI to measure how infants' brains respond to faces, bodies, objects, and scenes. And because infants don't really like it when you don't show them things, I used a curvilinear baseline rather than a fixation cross that's typically used in these experiments. And while it would be nice to measure infants' brain response as a function of experience, I can't exactly do that. And so instead, I'm going to measure the visual statistics of these stimuli to see if the visual statistics can give us any leeway into the organization of VTC, the responses in VTC. So from these categories, the scene condition had the most high spatial frequency information, which is what we would expect. But all of these conditions were relatively well matched for low spatial frequency content. The objects condition had the most rectilinear content, followed by the curvilinear baseline. And the curvilinear baseline and objects conditions had the most curvy content, with faces, bodies, and scenes being well matched. But I told you the hypothesis of the proto-architecture is that these feature maps are overlaid on one another. So for ease, I'm going to do a linear additive process of these two, of curvilinear and low spatial frequency content, to say that our face-selective response, or proto-face area, would initially respond similarly to faces, bodies, and scenes, and more to objects in the curvy baseline. Whereas a proto-scene area would have a high response to objects and scenes, a lower response to the baseline, and the lowest response to bodies and faces. All right, so our alternative hypotheses are that perhaps a proto-face response and a proto-scene response correspond to the low-level visual statistics of the stimuli. Alternatively, perhaps infants have high-level category selective responses, such that there's a face-selective response and a scene-selective response. So to ask this question, I again collected data. I'm telling you the amount of data I collected from 2 different experiments combined across COIL 2011 and COIL 2021. I can tell you more about those things later if you want. We recruited 86 infants for this experiment. All of the infants were between 2 and 10 months old. We had enough data from 52 infants for the group random effects analysis, and enough data from 39 infants for the ROI analysis to be included in the analysis. Again, we scrubbed out all of the high motion data and required there to be at least 24 consecutive low motion volumes and at least 96 volumes per subrun. And infants had to have at least 2 subruns to be included in the analysis. Again, we swaddled infants and gave them hearing protection, Coil 2021 is the one that had the size adaptive frontal paddles. And with these data, we're going to ask, is there evidence for a proto-face or face-selective response in FFA? So here are some maps from individual infants. Both of these infants are about 5 months old. These are representative subjects, so not the best activations, but also not the worst activations. And as you can see, the area highlighted in purple is where we would expect there to be a face response. And indeed, you see a face response relative to objects, exactly where we would predict. I also want to point out that they're not in exactly the same location for these two infants. And so we're going to do an ROI analysis across infants. Our parcel of interest in this case is going to be the fusiform face area, or FFA parcel, which I've highlighted in purple on an infant's brain here. And I'm just going to remind you, this is the predicted proto-face response based on the visual statistics of the stimuli. And just like a little note about the analysis, all of our responses are going to be relative to the baseline condition. So the baseline condition is going to be represented by 0. So I'll just replot our expected proto-face response in reference to the baseline condition. So as you can see, the object response here would be pretty similar to the baseline response, and the face, body, and scene response would be less. All right, so what did we actually find in infants? Here's the face response, plotted in purple. As you can see, it's significantly greater than the response to bodies, objects, and scenes, and also significantly greater than the curvilinear baseline response. So yes, our evidence supports that there's a face-selective response in infant FFA. So next, let's ask, is there evidence for a proto-scene or scene-selective response in PPA? Again, I'll start with showing you some individual maps from infants. The expected scene response is highlighted in green. The location of the expected scene response is highlighted in green. And as you can see, both of these infants have small but present responses to scenes relative to objects in the expected location. Again, we'll do an ROI analysis. Again, here's a reminder of what a proto-scene response might look like based on the visual statistics of the stimuli. And we're going to replot these responses relative to baseline to make a comparison easier. All right, so here's the scene response. Surprising to us and surprising to the visual statistics hypothesis, the scene response is not significantly different than the baseline response. However, it is significantly greater than the response to faces, bodies, and objects. So between these two hypotheses, the evidence supports that there is scene selectivity in PPA, but there is a question about what exactly that means. Okay, so I provided you with evidence for face selective responses in FFA, scene selective responses in PPA, and I suggested that these responses do not match what you would expect for a proto-architecture hypothesis. But some of you might be saying, well, you did a linear addition of these two things. So I'm going to put these out here without combining them so you can compare the results directly and still claim that the proto-organization hypothesis is unlikely given the visual statistics of the stimuli and the responses we observed in infant FFA and PPA. All right, so next I'm going to ask, does infant extrastriate body area respond selectively to bodies? Again, here's a couple of individual maps, and you can see there are significant activations to bodies relative to objects in the places that we would expect outlined in pink. We're going to do our ROI analysis again in EVA, highlight it here in pink again. Here's the response to bodies. And indeed, it's greater than the response to faces, objects, and scenes, suggesting that even in infancy, the EBA responds selectively to bodies. Great. So do infants have face, scene, and body selectivity in FFA and PPA and EBA respectively? The answer appears to be yes. All right, so next I'm going to ask if cortical face responses emerge in serial or parallel. So first we'll go back to the adult brain. The adult brain, as I've already told you, has a the FFA selectively responds to faces. But I didn't tell you that there's also a region called the superior temporal sulcus, or STS, that also has a response that if you measure it relative to bodies, objects, and scenes, the response to faces is higher. And a region called the medial prefrontal cortex, or MPFC, that if you measure it with respect to bodies, objects, and scenes, also appears to be face selective. But if you show subjects stimuli that vary in social content and then measure the response of these same voxels to things like happy faces, not happy faces, social agents or non-social agents in one experiment, or in a second experiment, stimuli that vary in social content but don't contain any faces, will those responses still care about faces only, or will they have some other content in there? So in the fusiform face area for the experiment that all had faces, you can see that there's a high response to all of the conditions, which is what we would expect because all of those conditions have faces. But the STS and MPFC have a differential functional profile such that there's a main effective condition, a main effective region, and a region by condition interaction. Now, what about those stimuli that had no faces but did vary in social content? As we would expect, the fusiform face area does not respond to any of them. But we get a dramatically different pattern of response in the STS and MPFC, indicating that these three regions, despite appearing to be selective for faces relative to bodies, objects, and scenes, do indeed have different functional profiles in adults. All right, so without going through all of the details of those stimuli, I'm just going to say, in adults, the FFA supports face perception and identity, the STS supports social perception, and the MPFC supports reasoning about self-relevant social partners. And now I'm going to ask, well, how does this face selectivity develop? A prominent hypothesis in the field is that infants are born with or have an early emerging face detection system that may or may not feed into a face perception system, and that may later feed into a social perception system. And then the latest emerging system will be something that helps you reason about other people and whether or not they're self-relevant social partners. I'm going to call this the serial hypothesis. Alternatively, it's possible that just as early as infants are able to detect a face, they are also able to evaluate that face in terms of whether or not it would be a self-relevant social partner. I'm going to call this the parallel hypothesis. Specifically, the serial hypothesis predicts that FFA face selective responses will emerge first, and STS and MPFC face selective responses will only emerge after FFA. Conversely, the parallel hypothesis predicts that as early as infants have face selective responses in FFA, they also have face selective responses in STS and MPFC. All right, so first we might want to just say, well, if we average across all subjects, are there any face activations in STS and MPFC? So that's what we did first. And indeed, here's our expected FFA response, and there's an STS response, and there's also an MPFC response. So we are eliciting some sorts of activations in STS and MPFC, but will these responses to faces be selective in the same way they are in adults? So again, in infants, we're going to look in the fusiform face area, which I told you supports face perception, the superior temporal sulcus, which I told you supports social perception, and the MPFC, which I told you supports social evaluation. This time, we're going to combine across the two data sets. So the FFA activation is going to look a little bit different. Here's the face response. It's still selective. So that's good. But what we want to know is, since we have this face selective response in FFA, will STS also have a face selective response, or will that not be present yet? So we next ask about STS, and indeed there is a face selective response. But again, the series hypothesis predicts that FFA and STS might come before MPFC. So even though we're seeing it in FFA and STS, we might not yet see it in MPFC. But indeed, we do see a face selective response in MPFC as well. But for those of you that know anything about babies, you'll say, but that age range is pretty large. There's a lot of learning that happens between 2 and 9 months. So next we asked if we can find any evidence for age-related change that would suggest that one region emerges before another. And for this, we're going to just do a simple age correlation analysis. I'm going to plot age that's been z-scored on the x-axis and the contrast value on the y-axis, where the contrast value is response to faces minus the average response to all of the left out conditions. We do not see an effective age in FFA. What about STS? Again, no effective age. And we don't get an effective age in MPFC either. But you might be saying, well, you're kind of underpowered to be finding an age effect. You only have 39 infants. What if you just look at only the youngest infants, those that are 2.5 to 4.6 months old? So we did this next. We looked at just the youngest infants. And we had 16 of those, and in FFA, STS, and MPFC. So here's the response to faces in FFA. So far, it's the smallest response to faces we've seen yet, right? And it's still greater than the response to bodies, objects, and scenes, but it's not quite as big a difference that we've seen so far. So maybe this will be where we're going to find evidence for serial development, right? So let's look at the STS next, which shouldn't be there. Just a reminder. But it is. Look at that. We get a face-selective response and STS too. Okay, but maybe infants understand something about social perception, but they can't yet like evaluate people. So let's look at MPFC. Wow, there's a face-selective response there too. Okay, so this is definitely evidence against the serial hypothesis and in support of the parallel hypothesis. So that's kind of cool. So I asked, do cortical face responses emerge in serial or parallel? And I provided you with evidence that these face responses likely emerge in parallel. So now I'm just going to give you a few thoughts on the emerging mind. So when all of you look at these pictures, you're doing a lot of different things and you're making a lot of rich inferences. You may reason about what's happening in the pictures, what the people are doing, what their goals might be, and even the nature of their relationships. Surely infants are not able to make the same rich inferences that we do. At the beginning of my talk, I told you that what I really want to understand better is what the starting state of the human mind is. Sorry, Hannah's enjoying the pictures, so I just want to stay a little bit longer. I'm looking at a specific Julie right now because she got this picture taken of us and I think it's just so beautiful. And one of the privileges of being here and having such lovely people. But anyways, when an infant sees this picture, do they merely see two faces or do they also reason about the relationship between these two people? So I'm gonna talk a little bit about my ideas about this now. This is highly not based on very much data. It's just a bunch of thoughts that I've been thinking. And I'd love if you guys ask me questions about these things. But I'm going to talk about knowledge and suggest that primitives are the most basic unit of knowledge that support learning. And I'm going to talk about learning, which I'm going to define as information acquisition and integration. All right, under the knowledge side of things, there have been historically empiricists have suggested that infants are born with no functional structure, they have simple sensory content, and no built-in knowledge. Whereas nativists, on the other side, have postulated that infants have rich structure in their mind, lots of conceptual content, and a lot of built-in knowledge that's usually abstract. Empiricists suggest that the primitives that infants have available to them are low-level sensory primitives, which in the visual domain would be things like spatial frequency, lines, curves, and the auditory. I've talked to you already about all of these things. But nativists would posit that there's rich conceptual structure, and infants have knowledge about things like agents, objects, and even social relationships. Today, I showed you evidence that infants have face-selective, body-selective, scene-selective responses, and that they might also have music-selective and speech-selective responses. So I suggest that we, on the empiricist side of things, perhaps we should change our framework to be perceptual, such that the most basic unit over which infants are able to learn are things like faces, bodies, and scenes in the visual domain, and things like music and speech in the auditory domain. And that rather than thinking about no functional structure, empiricists might want to acknowledge that there's some minimal functional structure in infants' brains that include high-level perceptual content. Now I'm going to focus specifically on faces, because I've talked to you more about faces than anything else so far, and ask, do infants' brains prioritize faces to support learning, or do abstract concepts about people and agents support learning? I showed you that adults have 3 regions that are face selective: the STS, FFA, and MPFC. And I also showed you that these regions are also face selective in infants. I then showed you that these regions support different functions in adults, and suggested that because they support different functions in adults, they might also support different functions in infants. But that might not be true. So one hypothesis is that infants have no abstract knowledge about people, and that faces guide learning. So this would be on the empiricist side, but suggesting that perceptual content is faces, is the most fundamental primitive. And the implications for learning here are that infants learn that faces are more than a perceptual object, and that faces are attached to people that have goals and relationships. This would predict a relatively slow trajectory, and I think lots of people that are on the empiricist side of things would be happy with something like this. The implications for the brain are that infants' cortical responses to faces will not differentially represent social content, just the presence or absence of a face. And that cortical representations that are initially perceptual will develop into richer concepts with more experience. Alternatively, it's possible that infants have abstract knowledge about people and agents that guide learning. The implications for learning of this is that infants come to learn that faces are an important perceptual cue because they provide important information about people and agents. And the implications for the brain is that brain regions that represent people and agent knowledge will respond more to faces when compared to bodies, objects, and scenes. And it suggests that functional organization is present early and supports within domain learning. But if you're like me, you might be wondering, well, what mechanisms could be guiding this kind of learning? So developmentalists have thought about learning a lot. Some developmentalists talk about statistical learning, suggesting that babies primarily learn through statistical associations. Whereas another group of developmentalists primarily prioritize rational learning, suggesting that infants have rich structured learning and are able to make logical inferences over a hypothesis space. There's also a third kind of learning that I haven't seen talked about in the literature much recently, but back in like the 70s was talked about quite a bit, which is reward. It's a reinforcement kind of learning, and it happens obviously in infancy, just like it does for every other organism that we've measured, where pleasure increases behavior in a reinforcement style and increases motivation. This kind of learning is supported by the striatum and VTA. And faces elicit activation in both of these regions. It's weak and hard to measure, but it's potentially present. So as a postdoc, I'm going to go work with Randy Buckner to learn more about the human reward network with hopes that I can bring that back to my infant work and better understand how infants incorporate knowledge about the world to learn. And so with that, I'll just leave you with this last thought. One thing I wanted to learn is how an infant like this, before she's even opened her eyes, has any information about the world whatsoever, how her brain helps her to become the amazing person that she is. And so with that, I'll say thank you. Thank you especially to my advisors, everybody that helped me, families. All right, so I'd really love some questions if you have them.
First of all, there's an open question section where anybody here can answer questions, and also Kirsten is keeping an eye on questions from the Zoom window for people here remotely. So feel free to ask any questions, and then after that, there will be a closed session with just the committee. And when that's done, we will send out an announcement for the celebration afterwards.
Vandana has documented much of my scientific trajectory. All right, so I can call on people. Oh, there we go.
Hi, thank you. Lovely work, fantastic. Congratulations on really, path-breaking work and really interesting. I just want to offer you an opportunity to expand a little bit on what you got at the very end regarding the role of reward and reinforcement.
Sorry, I had trouble hearing you.
The role of reward and reinforcement, and how in the further maturation and development of all of these functions. That's at the very end, and it seems like actually a very rich, very broad.
Yeah.
So where do you want to get that?
Yeah, that's a great question. I actually think that for infants, interacting with people is rewarding. And so I think that that's where a lot of their learning comes from, is by activating these reward mechanisms. And since reward systems are like phylogenetically pretty old, it seems like there's a lot of ways we can study that. But unfortunately, I don't know anything about the reward system. So that's why I'm going to go learn more about it in my postdoc. But I do think that, so for example, I can give you some data that's driven some of my ideas about this, but in the Romanian orphanages where infants have been like really deprived of a lot of social interactions, they still have fairly typical responses to faces. And I would postulate that that's because anytime they do get interaction with humans, it activates this reward system, and reward systems can be activated even if it's a negative environment, right? And so you can experience reward even if your overall environment is relatively abusive, right? And deprived of normal social content. And so I think that one way that they're able to develop these typical face responses is because they do get some interaction with humans. And even though it's a deprived interaction, it still elicits a reward response in the babies that drives them to attend more to these faces when they are there.
Possible hypotheses are that the reward itself is actually part of the actual development of the function actually needs reward.
Yeah.
Alternatively, reward could just be what gets the infant to the kind of stimuli and experiences that they need, and it's really those that drive it. So
I think that's totally true. I don't know how to test that yet. And so I'm hoping that like taking time to learn about the reward system will help me learn ways to test it. And looking in reward and processes in infants, there's a bunch of like reinforcement learning experiments from like the 70s and 80s that have some potential issues with them. But hopefully I'll get to do some more work on that. Thank you.
I'm speaking through the microphone so people on Zoom can hear. So I have some questions from people on Zoom. Jared Hicks first asked, although he had to go, if the difference in primary auditory cortex activations, finding the activation in some infants and not others, might be explained by how much or how deeply they're sleeping. For example, babies who are sleeping deeper might move less and thus have more data. And they can also have lower activations then.
Yeah, I think that's a great question. I don't think I can tease that apart in these data because I didn't measure sleep state. But I am doing a follow-up experiment to do the same experiment with awake infants. So hopefully at some future talk, I'll have those data.
And there was a follow-up question about whether you might compare this to data in adults, sleeping adults.
I have data from adults, so I can show people the data from the adults if they want.
Awake and sleeping.
Oh, awake and sleeping. Oh, it's really hard to get adults to sleep in the scanner. I tried really hard to tell adults to sleep in the scanner, but unless you combine it with the EEG to measure sleep state, and it gets really complicated, but perhaps at some point we will. More?
Yeah, I have one more from Matthew Belmont. Given your finding of parallel development in FFA, STS, and MPFC, is it right to construe these as separate functional regions or as nodes of a single social perception network that develops as a unit? From your existing infant data, can you extract causal information flow that might speak to this question?
Yeah, that's also a great question. I think, though I could be wrong, that FFA, STS, and MPFC are not part of the same networks. So I think it would be hard to construe these as results suggesting that they're nodes in the same network. But again, I don't know a lot about networks. And so I'm hoping to learn more about networks in my postdoc and have a better answer in a couple of years. Josh.
So I have two questions. Like maybe to some extent you already addressed this one, but is there more than two hypotheses?
Oh yeah.
Another one that Rebecca is more of a top-down hypothesis, right? Like, is it possible that your data are more consistent with that, or at least as consistent with that?
Yeah, it's definitely possible. But then the question, becomes, well, how is it that infants MPFC could recognize that it's a face without FFA recognizing that it's a face when FFA supports perception? And so I've been trying to think a lot about like what would be the neural mechanism that would drive something like that. And some people have hypothesized of a subcortical face template, which would be fast and direct the information straight to MPFC. And so maybe in the amygdala, or I forget where else, somewhere else. And so that is a possibility. I'm more akin to the idea of it being a reward mechanism, which again would be fast and drive that information to MPFC and support a top-down learning approach. But I think the difference between parallel versus top-down, we can't even begin to address with these data, given we found no effective age anywhere. And so that's going to take a lot more time.
Yeah, I mean, another possibility that there's a lot of behavioral evidence that face perception, the kind of thing that we think is more ventral street, like the social part, like the expertise in recognizing faces, for example, develops over a very long period of time, right? Decades.
Yeah.
So maybe would you say one possibility as well, sort of in place, the basic architecture is in place all from the beginning, but then as you get more experience seeing more people's faces, then there's a different developmental time course for that.
Yeah.
But you don't need to figure out who's rewarding or something. You just need them to recognize the differences between many different people.
Yeah, I think that's an interesting possibility that face perception is refined through development. And I tried to leave that as a possibility on my slide because there has been a lot of, I don't know the behavioral data as well as the neural data, but there's been a lot of neural data to suggest that FFA selectivity has a long protracted development. And I don't know the answer to that. I'm hoping to look at kids' FFA responses and see if I can find age effects using the same analysis that I did with infants and see if that's true. But one thing I would say though, is that even young infants differentiate faces in terms of like, that's my mom from like a few days after birth, and start attributing social meaning to faces in terms of like, that person is the same race or gender as my mom, but within a couple of months. And so I think the behavioral evidence also supports that a lot of face perception is really early emerging and doesn't take years of protracted development. And even like own race effect emerges pretty quickly. So it doesn't take us a ton of experience with faces for that to happen. And so I think that there's just this question of what does it mean for face selective responses to be stronger in older kids? And I'm not sure if that's a function of like neural development in terms of like myelination or how fast signals are sent, or even the ability of oxygenated hemoglobin to travel through the brain. And so I think those are all going to be interesting open questions. But yeah, there's a lot of hypotheses I think that these open up, but my hope is that they're starting to constrain some hypotheses that have been really prevalent in the field in terms of saying, okay, we can still believe in an empiricist approach, or we can still believe in a high-level conceptual approach, and both of those have merit, but maybe the empiricist approach needs to be constrained and stop talking about low-level visual features of images and start talking about categories that infants' brains represent. Does that make sense?
Yeah.
Okay.
I respect how you're trying to not weigh in on empiricists versus nativists, but is there a way you think the nativist approach needs to be modified?
Yeah. So I thought I was trying to think about how to do that well in this talk without getting too in the weeds, because I think about that a lot. And I think it was interesting the other day, a lot of people talk about intuitive physics, and that people, maybe infants, understand intuitive sociology and intuitive psychology in terms of, well, it's not intuitive physics, so it must be one of these. I would argue with Tomer or whoever has that I think intuitive sociology is probably the most fundamental knowledge that humans need to have, and other than like physical knowledge, and that perhaps if there is any innate human specific knowledge, it's going to be about other people, and that other people and relationships are important. And so I think one way we can update this is there's Rebecca has shown in her lab and in her data that even children as young as 3 have different networks that support reasoning about people's minds and versus supporting reasoning about the physical world. And I think that those two things are probably there in infancy, and studying how those develop and how they interact and talk with one another, whether it's through studying intuitive physics or intuitive sociology, I think will give us a lot of leeway into being able to refine those conceptual theories. Thank you. Haley.
Beautiful talk and beautiful work, Heather.
Thank you.
I think that one of your results that surprised me the most was the music selectivity response. And I was just curious if you could talk a little bit about what you think that means. And I also was trying to reflect on what you think about reward systems maybe kind of coming into play, and whether you think that might also impact the emergence of, or the existence of early like speech selectivity and music selectivity. Do you think all of those things are connected, or?
I actually would be surprised if, I don't know anything about reward in sleep. I'd be pretty surprised if there are reward mechanisms during sleep. I don't know, does anybody know? I don't know, anyways. So I don't think that reward is likely to be interacting with music and speech perception per se. I also thought the music response was surprising, but also exciting. I also thought the speech response was surprising and exciting. So I think there's a lot of open questions, but I think the first thing is to see, can we replicate this? And will it be any different in awake babies? So I'll tell you in a few years. Any more questions?
I have a question from Yibiao Liang asking about, given how young the infants are that you're studying, what are they actually seeing? Like how are they processing information such that they can make these categorizations of objects? I think it's a general question about the visual development process.
Is it about visual acuity specifically?
It seems like that's the point, yeah.
Yeah, so visual acuity is usually pretty good by about 8 weeks, and all of my infants are at least 2.5 weeks old. I mean, sorry, 2.5 months old. So above this 8-week mark where we would need to make sure that they have good visual acuity. But in addition to just saying, well, they must have good visual acuity, for every infant that I scan, I would go back to the back of the scanner board and put my head where the stimuli would be and play peekaboo with the infants to make sure that they could see me and I would get their attention so that I could be assured that they were actually able to see the stimuli. So I think that visual acuity is not an issue for these data. I think that's the question. So I'm going to leave that with that. Anything else? Yep. Okay.
So is 2.5 months like as is 2.5 months like as early as you think you can collect data from infants for fMRI?
No, it's the earliest I think I could get. I could be confident that I think they have good enough visual acuity, but for the auditory experiment, I don't care about the visual acuity. So I plan to do that in the same age range infants that I did for sleeping. And I'll be playing them a colorful stimulus to keep their attention. So to minimize head motion, but I don't care if they can actually see as long as it seems interesting. So it's fractals. Any other questions?
Okay.
All right.
In that case, we'll go on to the closed session and you guys will hear from us by email in a bit. Thank you.